You guys can see the power, like the full screen, right? Okay. So you can see this?
Yeah.
Okay, just checking. It's being weird. All right, before I get started, just want to share the discovery that I made yesterday, which is that Disney is sharing its churro recipe. So if anybody needs a good churro recipe, they have posted it on their blog.
Nice.
And apparently they just posted their Dole Whip recipe. So if anybody has a lot of pineapple, I think you can make that too. I didn't look because I don't have any pineapple, so it doesn't do me any good. Okay, so on that note, so nervous system is by far the most complex system in the body, like just period. And so we spend a lot of time on it, and honestly, even as much time as we're going to spend on it, we're barely going to crack the surface. So with that being said, the nervous system tissue chapter, what we're going to cover today, is like super long. And I tried my best to streamline it, and I tried my best to, you know, limit what we covered and to make it like a compact lecture, and it's still ridiculously long. So if we get to that point where we're like, this is too much and I can't talk anymore or whatever, we'll just roll it over to next week. But at least we'll get it started. The nice thing is that some of this is building on and even the same as what we talked about for muscle physiology. So one of the reasons it's nice to do the muscular system first is that all the electrical activity that we're about to talk about, we covered some of it already. So keep that in mind as we go through this, because some of this stuff really is just repeating and expanding on what we already covered. So these are the learning objectives for the whole thing. So when we covered nervous tissue, we're talking about the cells of the nervous system, their anatomy, and then we're talking about their physiology, as well as the overall functions of the nervous system and the overall organization of the nervous system. We're going to start with an overview of the overall organization so that you get a better feeling for where these cells are as we talk about it. So we're going to go over divisions and overall functions first, and then we're going to start breaking down the tissue by cells, anatomy, and physiology. So first of all, the nervous system is our major control system. We're not going to talk about the endocrine system in this class, but the other control system of the body is the endocrine system, which you should cover in the second semester of anatomy and physiology. They work together to basically control the functions and homeostasis of the whole body. The nervous system, though, is by far the one in charge. So when we say it's the master controlling and communicating system of the body, we do mean that literally. And the cells of the system communicate both by electrical and chemical signals. Now, the way this works is very fast and very specific. So the nervous system is very good at targeting whoever it's controlling. And that's because of its ability to have synapses. And we saw one of those already because we saw how the nervous system is able to control muscle fibers with those neuromuscular junctions. And because of the excitable nature of neurons, of the cells of the nervous system, they're able to have very rapid responses to stimuli. And we're going to start seeing how that works today. The nice thing about the nervous system is that even though it's very complex, we can break its functions down into 3 parts. Every single function of the nervous system falls into one of these 3 categories. So it's either going to be sensory input, integration of information, or motor output. Sensory input is how the nervous system has information to know how to act to control the body. So it makes use of sensory receptors, which we'll talk about more in the next week or so, which allows it to monitor and be aware of changes both inside and outside of the body. So all of the sensory input that you're aware of and a lot of it that you're not aware of is all coming into your nervous system. Everything you see, hear, taste, touch, all the pressure, temperature, pain information coming in from your skin, as well as a bunch of information about what's going on inside. So we talked about a couple of those when we learned about feedback loops. So like thermoreceptors monitoring your body temperature, right? We learned about that. We touched on chemoreceptors and baroreceptors monitoring the chemical status of your blood and your blood pressure. Those are internal sensors monitoring changes. So all of those things cumulatively together are called sensory input, because we're literally putting information into the nervous system. So that's one aspect of the function. All that information then goes on to the next function, which is integration. So integration is how the nervous system processes and then interprets all of the sensory input. It does a lot of correlation as well. So, you know, it'll take information from one sense and integrate it with another. And then it makes decisions about what to do based on that input. So, you know, some of the negative feedback loops we talked about before qualify as that kind of integration, where then the motor output is the response. So this is the major function of the nervous system, and it's the part that's super complicated. Luckily, it's also the part we're gonna not spend as much time on, because that's like higher level nervous system study. Once the integration function makes a decision, it goes to the next function, which is motor output. So motor output is how the nervous system controls the rest of the body, and it does it by activating what we call effector organs. So effector organs are parts of the body that have direct connections to the nervous system and are able to respond to their nerve impulses. And this is all of the muscles. So this is all kinds of muscle tissue. This is skeletal, there we go. This is skeletal, cardiac, and smooth muscle, as well as glands of the body. So these are things like what's going on? These are things like salivary glands. This is like your tear glands. Sweat glands. Anything that secretes anything is going to have some sort of control by your nervous system. And so that's how we get responses, which we call, of course, cumulatively motor output. Okay, so sensory input, integration, motor output. When we look at how we divide up the nervous system structurally, we see that there are two major divisions. So there's the central nervous system, which we abbreviate as CNS, and that is composed of the brain and the spinal cord. You can also think about it as everything in the dorsal body cavity. So that's the cranial cavity, literally the inside of your skull, and then the vertebral cavity, which is that whole space created by all those vertebral foramen that you learned about in the skeletal system. All other nervous tissue in the body is the peripheral nervous system, so-called because it's on the periphery of the central nervous system, so it's outside of the dorsal body cavity. And that falls into the following categories, and we'll learn more about these guys later. But cranial nerves, spinal nerves, and ganglia. And I promise I'll explain what all that means more as we keep going. So CNS and PNS. And here's a diagram of that. So central nervous system is brain and spinal cord. Everything else is peripheral. Now, that being said, the central nervous system has the major role. So that second role of integration that I talked about, the ability to process sensory input and then send motor output, that's the job of the central nervous system. So integration is what they do. Because their job is integration, the peripheral nervous system does the other two things. So the peripheral nervous system is in charge of both sensory input into the CNS and carries all the motor output. Because being outside of those dorsal body cavities means that the peripheral nervous system is the connection between the central nervous system and the rest of the body. Give me 1 second. I have to go tell the dog that there's nothing to bark at. I assume it's the male. All right, so that means that we can divide the peripheral nervous system into a sensory division and a motor division. And both of those are further subdivided into a somatic and a visceral. So somatic means body, but it actually refers to basically the musculoskeletal and integumentary systems. So basically all of the systems we've already talked about get controlled by the same division of the body, and also all carry sensory information in the same division. All the other body systems, the ones that we haven't learned about yet, those are all carried by the visceral division, both sensory and motor information. Now, the other term for this on the motor side is autonomic. So the motor divisions carry all the motor output to muscles and glands. Now the somatic only carries motor output to the skeletal muscles. So this is also all of the voluntary motor control, although not all of it is voluntary. And then the visceral side is what's also called the autonomic nervous system, so the visceral motor division. And that carries motor output to everything that's involuntary. So that's our smooth muscle, our cardiac muscle, as well as any glands. So here's a diagrammatic view of that, including showing the flow of information. So all the control is up here. This is where our integration occurs. And as we talk about various things, it's always going to come back to these three functions: sensory, integration, and motor, okay? So the sensory division is going to carry sensory information from the peripheral nervous system to the central, and then the central sends motor information out through the peripheral, and where it goes depends on what's being controlled. So somatic is skeletal, oh boy, and autonomic is everything else. And as we'll talk about more later, the autonomic nervous system has a further subdivision, and we'll cover that in another couple of chapters. So just so that you've got it all in words as well, so that it's really nicely broken down. So the somatic division of motor output is, and this also does apply to the sensory too. So conscious perception is awareness of the sensory information. And then voluntary responses, as well as control of skeletal muscle that is involuntary. So that includes things like reflexive actions, as well as things like, you know, like when you're walking, but you're not actually thinking about the walking part and it's still happening automatically, that's still the somatic nervous system. And then everything else is visceral. So most of the sensory input here is unconscious, although we are aware of some visceral input. So, you know, like we know when we're hungry, for example. We know when, you know, we know when something in our digestive system is upset and we have pain from it, like we still are gonna feel that. So there is a little bit of crossover between what's conscious perception. They're also going to be in charge of all the homeostatic processes. So all of our negative feedback loops and positive feedback loops for the most part. This is always going to be involuntary. We do not have direct control over anything on this side. And again, it's smooth muscle, glands, and cardiac muscle. Although as you'll learn when you get to the cardiovascular system, it's sort of an indirect control. Like it's control, but it's not, the nervous system doesn't control the heart's ability to beat. It just influences how fast and how strong it beats. So don't want you to think that your brain is actually controlling your heart, it's just influencing it. And then here is another chart of the divisions and roles of the nervous system. And so this is showing you the two parts of the central nervous system up here and what they do, and then the parts of the peripheral nervous system. And then it's got another part that we're not going to talk about in the nervous system. We're going to save this for the digestive. So the digestive system actually has an extensive organization of nervous tissue in it. We call it the enteric nervous system, or ENS. And it has a lot of control over the function of your digestive system. And while it's in communication with your central nervous system, it can do a lot on its own. So even though it's technically part of the nervous system, we're going to talk about it when we get to the digestive system, because that's where it's actually having its effects. All right, any questions about our overview before we get into our actual nervous tissue? All right, okay, so nervous tissue. So this is our fourth type of adult tissue, right? We've talked about epithelial tissues, we've talked about connective tissues, and we've talked about muscular tissues. And now we're finally at nervous, and it's totally different from the other guys. So this is really just about different types of cells in nervous tissue and how nervous tissue is organized. So the cell types in nervous tissue are neurons and glial cells. Neurons are the primary guys. They're the ones that carry out the communicative role of the whole system. And that's because they're the ones that are electrically excitable, which means that just like muscle tissue, they're capable of responding to stimuli. They also have conductivity. So just like electrical wires, they are capable of conducting electricity, although they do it in a slightly different way. And the other thing that they do, and this is the one that's different from muscle tissue, because technically muscle tissue manages this too. What they do that's different is secretion. So secretion is how they're able to spread chemical signals by producing and literally excreting or secreting chemicals. And that's, of course, neurotransmitters. We learned about one already at the neuromuscular junction, acetylcholine. So that's when we talk about secretion, we mean neurotransmitters. Now the glial cells are the support cells for the neurons. And we have 6 types total. They all have different functions, and there's different ones in the central nervous system compared to the peripheral nervous system. So we'll talk about each one, what they do, and where they're located. And we'll get into more detail on the neurons as well. So a couple things about neurons. Neurons have a very unique anatomy. So they have a cell body, which we typically call the soma. So again, soma means body. And then they have cellular extension. So they have things coming off of the cell body, and we generally refer to them as processes. And they have an axon, and we'll talk about this more later, but they have an axon, and then they have dendrites. Axons send information, dendrites receive information, but collectively we call them processes, okay? Because they stick out. Okay, now we find that neurons are organized in such a way that the cell bodies typically are hanging out together in the same regions, both in the CNS and the PNS. And those regions have a different color than the regions where the processes are hanging out. So we call the regions with neuron cell bodies gray matter. And then the axons of those same neurons are running together. They can be very, very long. So they're running together in pathways. And there's a lot more lipid in those areas for reasons that I'll explain shortly. And it makes it look white, so we call those areas white matter. Now, you've probably heard about gray matter and white matter before. It comes up in common conversation sometimes, especially in regard to the brain. And I don't know why I put this here, but I did, so I'll say it. So the pathways of axons in the central nervous system are called tracts, and the pathways of axons in the peripheral are called nerves. So like I said, this is all about being very, very organized. So in the brain, for example, we have a superficial layer of gray matter, and then there's more deep inside as well, but this is gray matter. It doesn't always look gray. In really fresh nervous tissue, it's gonna be more pink, and that's actually because there's a lot more blood vessels in those areas. And then by contrast, where all those axons are running looks much whiter, because again, it's got a lot of lipid in there, and the lipid makes it look white. The other thing is that gray matter in the central nervous system, we call them nuclei. So a collection of gray matter that's deeper in the brain or that's in the spinal cord is called a nucleus. And then in the peripheral nervous system, it's called a ganglion. So I said the peripheral nervous system includes ganglia, that's plural, and it means gray matter. And the other things I said for the peripheral were nerves, so that's white matter. And your book thinks it's important, and it is good to kind of point out that this is another area of, we're using the same word again to kind of mean the same thing, but not. So nucleus has three major meanings. It can refer to the center of an atom. It can refer to the organelle containing DNA in a cell, and it can refer to a collection of neuron cell bodies in the central nervous system. So they made this nice little comparison so that you've got that down. So if you're thinking nucleus sounds awfully familiar, I should hope so, because we've talked about it several times already, okay? So clearly nucleus just means, you know, the central core of something. And so we've used it in multiple situations where it has that meaning. All right, so details about neurons. So you might also see these guys called nerve cells, but I try to stick with neurons. Now they are the structural and quite frankly the functional unit of the nervous system, because a single neuron is capable of receiving, integrating, and sending messages. So they are capable of all of the functions of the entire nervous system, although it does technically take more than one to actually get something done. So characteristics of neurons, and this is kind of compared and contrast with characteristics of muscle cells, and technically with connective tissue and epithelial cells. So they are excitable. They share that with muscle cells. And the electrical response to stimulus is nerve impulses. Neurons tend to be very long-lived, so they have what's called longevity, and they're often able to last a lifetime. Technically, that is also true of things like skeletal muscle cells. They're fairly long-lived too, but we never really focus on that. We do focus on it for neurons. They are amitotic, so they're unable to divide. Again, this is not actually unique. A lot of cells that we've talked about are not able to divide once they become fully specialized, right? So a lot of our specialized connective tissue cells like osteocytes and chondrocytes, you know, we know that our epithelial cells will sometimes lose the ability to divide, for example, in our stratified tissues. Our skeletal muscle fibers definitely can't divide anymore. But it seems to be more important in neurons because they also typically lack a stem cell supply, so they can't regenerate if they're damaged at all, like we can't replace them. We used to think that that was just a hard rule, like there's no replacing neurons, but now we know that there are some places where they can be replaced. So that was definitely good news when we started figuring that out. And we're still learning more about that. The other thing that they have, which they have in common with muscle, is having a very high metabolic rate, because there's a lot of ATP that goes into all this electrical activity and secreting of neurotransmitters. And so they have, they require a continuous supply of oxygen and glucose. And that means that they're always very vascular, because you need a good blood supply if you are going to have a good supply of oxygen and glucose. Hold on. All right, so really good blood supply so that they always have oxygen and fuel for ATP. And as I already mentioned, their anatomy is super specialized, very much not a typical cell. And we do divide the whole structure into two parts, the cell body and the cellular processes. And then the other thing that's very important and special is their ability to form synapses, which are chemical or electrical connections with other cells. And we'll talk about that more later. So here's a typical neuron. So we've got a cell body here, and then all the stuff coming off is the processes. So the cell body has a couple of different names. I already said soma. Perikaryon is another one. I will probably never use that one again. It's a mouthful and it's awkward, but just so that you're aware that there's another name there. This is where not only the nucleus, but also the majority of the cytoplasm and the organelles are located. And because of the neurotransmitter production that's going on, there's a lot of endoplasmic reticulum. So especially rough endoplasmic reticulum, and it's typically really dark. And so they gave it a name, which is Nissl bodies. Again, not gonna ask you that, probably never mentioning it again, but it does come up sometimes when you study neuron anatomy. In order to maintain that very unique shape that neurons have, they have specialized cytoskeletons. So intermediate filament bundles called neurofibrils help maintain that shape. And the cell body makes proteins and any other chemicals that the cell needs, so including neurotransmitter. And then it transports the neurotransmitter all the way down the cellular process of the axon to the axon terminal, because that's where it's going to be released. So that's where the synapse gets formed. The vast majority of cell bodies are located in the CNS, not in the PNS, okay? So central, not peripheral. And as I said, we call those groupings nuclei. So that's all in here. And this diagram doesn't show you the organelles and the cytoskeleton and stuff, but it's in there. You know what all that stuff looks like. So then the processes come in two types. So dendrites, which just like means branches, are variable. So some neurons might actually have no dendrites. Most of them have multiple dendrites. Most of them are short, branching, and they kind of surround the body of the cell. And their job is to be an input region. So this is where the neuron receives information. Receives information. So synapses do form on dendrites, but it's the receiving side of the synapses. And those locations are called dendritic spines. Again, details that I won't really ask you about, but just so you know. Okay, so dendrites are the receiving side. Axons are the output. So neurons always have one and only one axon. The beginning of the axon as it comes off of the cell body is called the axon hillock, because it's like a little bulgy, I guess. And although there's only one axon, it is allowed to branch. So axons can branch. We call those axon collaterals. Usually they're near the end of the axon, so we call them terminal branches. And then the very end of the axon ends in little knobs, which you've seen before because the axon terminal was what we saw at the neuromuscular junction, right? Forming the synapse with the muscle cell. So axon terminal. And we also are familiar with the fact that there's branches because that's how we can have a motor unit, right? So we can have one neuron controlling multiple muscle fibers. Oh boy. Because of this terminal branching. So this is where the nerve impulse, the electrical activity, gets conducted all the way down the axon, and then we get chemicals released across the synapse. And we'll talk about the physiology when we get through all the anatomy. Okay, so this is what dendrites look like on most cells, short and very, very branchy, allowing for a lot of connections coming in, okay? And then there's always one pathway of communication out, although it can branch at the end to synapse with multiple cells. And we're going to talk about whatever's going on here in a little bit. So, just like everything else, there are classifications for neurons. So we can classify them by function, and we can classify them by structure. And there are 3 types in both categories, or classification schemes, I guess. Now the functional, we'll talk about second. But I want you to notice that it's the same 3 functions as the whole nervous system, okay? This is going to be a theme through the whole thing. Motor, integrative, sensory. Structurally, it's based on the number of processes seen coming off of the cell body. So the typical one that I showed you before is called a multipolar neuron because it's got multiple dendrites and, of course, the single axon. Now, sometimes, even though there are still dendrites coming off, the organization off the body is a little weird. So you can have a bipolar neuron where you have a single axon and a single dendrite coming off from either side. So bipolar, 2 poles. Or you can have a unipolar where there's really just one thing coming off. Now we also call these pseudo-unipolar. So remember, pseudo means fake. So it looks like it's only one, but really there's still dendrite on one side and axon on the other. Okay, so 3 types of structure, and then they actually do pair up pretty well with the functional classifications, not unlike our joint classifications. Just so you know, even though there's just the 3, we do actually have some specialized ones. They're kind of technically multipolar, but they're so weird and so unique looking that they were given their own names. And you don't need to know these guys, but I just want to kind of make you aware of them, and we'll mention them later on when we get to the particular things that they do. So this, for example, is called a pyramidal cell because it's kind of like a triangle. We see these in the cerebral cortex, which is that outer layer of gray matter that I showed you on the picture. These guys are really important in all the integrating functions that we see. This guy is called a Purkinje cell. Some guy named Purkinje discovered it. We still haven't changed that name. And it looks a lot like a multipolar, but the dendrite situation here is obviously excessive. And we find those in another part of the brain, and we'll talk about them later. And then we're going to talk about these guys. These are part of our ability to smell. These are olfactory cells, so we'll get to them later, but they're basically bipolar. And I really don't know why they threw that in there. Okay, so functional classifications. Remember, it's sensory, integrative, and motor. So we can have sensory neurons. We also call them afferent neurons because afferent means that you're sending information into the nervous system, into the central nervous system. So sensory neurons are in charge of collecting sensory information and sending it into the CNS. These are the only type of neurons whose cell bodies are actually in the peripheral nervous system. Wow. No, they're not the only ones. This is one of the types. This is one of the types of neurons whose cell bodies are in the peripheral nervous system. And so we call those locations sensory ganglia. So remember, a ganglion is a collection of cell bodies in the peripheral nervous system. And these are all sensory, so sensory ganglia. And these are almost always unipolar. And when we get to the anatomy of the peripheral nervous system, we'll see how that makes sense. Okay, so sensory input, unipolar. Interneurons or association neurons are what we call neurons whose job is to perform the integrative and processing functions of the nervous system. These guys are always in the central nervous system, so they're always in the brain and the spinal cord. And their job is to make all of the processing connections between the sensory neurons and the motor neurons. And this is such a big task that 99% of the neurons in our body are interneurons. So inter means between, so literally they were named for being between sensory and motor neurons. And the vast majority of them are multipolar, which means that most of the neurons in the body are multipolar, okay? I'm just doomed to bark. I swear. Okay, hold on. Hopefully that works. I don't have to get up. All right. So most of them are multipolar interneurons. So that leaves us with motor neurons. And motor neurons are also called efferent. So efferent things carry stuff away from the CNS, and effectors are what we call the organs that respond. So this is the muscles and glands. All right. Afferent in, efferent out. So motor neurons are responsible for motor output, and these also are usually multipolar neurons whose bodies are in the central nervous system. There are some exceptions, which is why I had to change my statement earlier about sensory neurons being the only ones in the PNS. There are some exceptions, and we'll get to them later. We're not going to worry about it right now. So yeah, most neurons are multipolar. Most neurons are integrative or interneurons, and most of them have their cell bodies in the central nervous system. Any questions about neurons before I move on to glial cells? No? Okay. So one other thing I want to mention before I forget. So what that means too is if most neuron cell bodies are in the central nervous system, it means that when we divide up the nervous system by central and peripheral, the peripheral nervous system is mostly axons. It's mostly motor axons and sensory dendrites, which is kind of weird. So we're actually dividing up the system like right down the middle of cells because they're really, really long. Okay, so we'll tackle that more when we get to next week. All right, so then other type of cells is our glial cells. Neuroglia is the other term for it. And these are described as the support cells of the nervous system. So they do everything else to keep the system functioning so that the neurons can do the communicating part. And like I said, there's 6 total cell types, but they're divided. So there's 4 in the CNS and there's 2 in the PNS, okay? Now, in case I forget to say it later, these 2 in the peripheral nervous system do the same jobs as some of the cells in the central, okay? So satellite cells and astrocytes basically do the same thing. Oligodendrocytes and Schwann cells basically do the same thing. And as you'll hopefully see as we go through this, the PNS doesn't need these other guys, and that's why there aren't equivalents, okay? So we'll start with the CNS types. Astrocytes are arguably one of the most important glial cells. So they were named because they kind of look like stars. This is a picture of one from Wikipedia. As you can see, they have extensive cellular processes that are neither axons nor dendrites. They're a whole other type of cellular process. And their job is to control the environment around neurons. So they control the exchange of materials between neurons and their blood supply. And that lets them control entirely the chemical environment around neurons. Now we'll talk more about how they do that later when we get to details about the central nervous system. But basically that makes them super important, okay? And we're pretty sure they have even more jobs than that, but we're just starting to figure that out. Second one is microglial cells. They're called microglia because they're the smallest ones, and they are the immune cells of the central nervous system. So they monitor neuron health, they phagocytose any invaders, as well as cleaning up damaged cells after any injuries or infection. So they act very much like macrophages. We think they might originally have been macrophages, and certainly their role is quite important for protection. Okay, so microglia, very, very small. Some of these guys in this picture are microglia. I can't remember which ones, but we'll just go with these guys. Third kind, ependymal cells. Ependymal cells have very specific function and location. So they line the cavities in the brain and the spinal cord, and they produce and circulate the cerebrospinal fluid within those cavities. So we'll talk more about CSF later, For now, all we need to know is that the CSF acts as a cushion for the brain and the spinal cord, so it has a protective function, and that the ependymal cells look very much like ciliated columnar epithelial cells because they have cilia, and the cilia function to circulate the fluid. And we'll talk more about CSF production and composition and additional functions when we get to CNS next week. Okay, so ependymal cells. The last ones are oligodendrocytes. Oligo means like few or several, and that refers to the number of processes they have, which look like dendrites but are not. And they use those processes to wrap themselves around the axons of the neurons, should really say neuron, in the central nervous system. When we have a cell wrapping around axons, we call that myelination, which we'll talk about as soon as we're done talking about glial cells. So basically, like, there's an oligodendrite cell body here, and it's got some processes, and it wraps around the axons. And I'll show you better pictures later. Okay, so 4 types of glial cells in the central nervous system. This is kind of what diagrams usually look like for that. So we've got a neuron here. It's got astrocytes controlling it or protecting it or whatever. Microglial cells hanging out ready to do their thing if needed. Ependymal cells down here, kind of showing you that they are in communication with the other types of glial cells, and the CSF would be over here. And then here's some oligodendrocytes with their processes wrapped around segments of axon to myelinate them. Okay, so those are our 4 types of CNS glial cells. And then we've got the 2 types in the peripheral nervous system. So the peripheral nervous system doesn't have cerebrospinal fluid, so there's no fluid, so there's no need for ependymal cells. And the regular immune system is able to access it, so there's no need for microglia. So all we need is cells to control the environment around the neuron cell bodies, and that's what our satellite cells are for. So they surround the neuron cell body, just like a satellite orbits a planet kind of a thing. And although they probably don't do exactly the same things that astrocytes do, we're pretty sure that they basically do the same general controlling of the chemical environment. And then Schwann cells do the same job as oligodendrocytes. So they myelinate axons in the peripheral nervous system. And then even though not all axons are myelinated, they still surround all of them and basically help them stay structurally supported and organized inside of nerves. Now, Schwann, I believe, was a person. Like, it was definitely one of those names that got named after somebody or someplace. The newer term is neurolemocyte, but this is one of the rare instances where the new term sucks because it's a mouthful, and so we still use Schwann cell, even though neurolemocyte is probably a better name, more accurate. Okay, so those are those 2 guys, and we got a picture here. So here's some Schwann cells wrapped around some axons, and then satellite cells wrapped around the cell body. And as it points out here, this is a unipolar sensory neuron. So the cell body's here, the single process coming off is here, and then it's running both ways as an axon. And over here somewhere is the dendrite. So let's tackle this myelination a little bit more. So myelination is insulation of the axon, and it is electrical insulation composed of the plasma membrane of the cell doing the myelinating. Axons are covered in segments with gaps in between, and when we talk about action potentials, we'll see that the gaps are very important. And the reason why axons are myelinated is because it increases the speed of the transmission of the electrical activity. So it increases nerve impulse speed. And of course, as we just said, different cells myelinate when you're in the CNS versus the PNS. So oligodendrocytes versus Schwann cells. So this is a diagram of a Schwann cell myelinating an axon. So it basically engulfs the axon and then wraps its membrane over and over and over again, shoving the cellular contents of the cell to the periphery or the outside. So here's the nucleus and the cytoplasm, and then we have layers and layers and layers of plasma membrane. And this plasma membrane surrounding the axons is why white matter looks white, because it's the phospholipids of the membrane. This is an electromicrograph of a myelinated axon, and you can really see how many layers there are here. Like, it's really impressive. And then, let's see, I think either that or maybe that. Somewhere over there is the nucleus. It's really hard to tell. But anyway, and then the axon's going to be in here. And then here's a diagram comparing. So this is a myelinated axon in the peripheral nervous system with Schwann cells, individual, one cell per myelination segment. And then this is myelinated axons in the central nervous system. As I showed you before, the oligodendrocyte sends out a process, and that does one segment. So it usually does a couple. And then this is an unmyelinated axon. So it's a naked axon right there. Okay, any questions about the anatomy before we move on to the physiology? All right, so let's talk action potentials. Now, the nice thing about this is that it is basically review, okay? So I'm gonna take you through this, remind you of the things you've already learned, and just kind of show you how it applies to neurons as opposed to muscle cells. So we're gonna go back through all the things required to have a resting membrane potential, as well as to have an action potential, and what that looks like as we go through it. So the keys here are the same as they were before. So different kinds of ion channels, having a resting membrane potential established by the sodium-potassium ATPase or sodium-potassium pump. So relevant types of channels. So ligand-gated channels. So this is, of course, the channels that are able to bind neurotransmitter to open. So we find these on the receiving side of a synapse. And we'll talk about them more later. Not super relevant for action potentials immediately, but we'll definitely be getting to them later. Mechanically gated ion channels are going to be relevant for sensory neurons, and we'll talk about them more next week. This is where the picture is, so I threw it in, okay? So basically, these are ones that open in response to mechanical changes to the membrane. So this is for stuff like touch, pressure, vibration, things like that, okay? So we'll get to that later. And then, of course, our most crucial type of channel for action potentials is our voltage-gated ion channel, which, you know, is going to open in response to changes in membrane potential, and typically it's going to respond to some sort of depolarization, or becoming less negative. And then the last guy is our leak channel. So remember, leak channels are always open, or according to this, randomly open. But anyway, there's nothing in particular controlling whether or not stuff is flowing through. And the big leak channel that we see relevant here is the potassium ion leak channels, because they work with the sodium potassium pumps to maintain that slightly negative environment inside of the membrane for resting membrane potential. So, sodium potassium pump, right? Using ATP to keep potassium inside of the cell, to keep sodium outside of the cell, and that's going to regulate the charge inside and outside the membrane and maintain membrane potential. So as long as we keep potassiums in here, sodium's out here, and then we're letting a little bit of sodium leak out, even as we're better at pumping, or sorry, a little bit of potassium out, even as we're better at pumping sodiums in, because remember, the pump gets 3 sodiums back out for every 2 potassiums that it brings back in, and that's how we get that nice little negative charge. It's a combination of the disparity between the sodiums and potassiums with the pump and a little bit of the leak channels allowing potassium or positive charge out. Most of our neurons have a resting membrane potential of negative 70 millivolts. Not that you need to know that number. One second. Oh my God. Nope, I'm okay. All right. So there's actually 2 different types of changes in membrane potential that we can see in neurons. There's graded potentials and there's action potentials. Now, we've talked about action potentials before, but we haven't really addressed graded potentials. So graded potentials are short-acting signals. We usually see them on the receiving end of synapses. So neurons that are receiving signals will have these small, very localized changes in membrane potential. They actually decay with distance. So they typically don't depolarize the entire membrane, just a little segment of it. And we'll see how that works over the next couple of slides. And we can compare that to the action potentials, which are long distance. They're able to travel the entire length of an axon, and they maintain the same signal strength the entire time. So we call them all or none, which I'll talk about more in a second. All right, so here's our classic action potential diagram, right? So this is voltage, and this is our resting membrane potential. This is a stimulus that slightly depolarizes to threshold, which is when our voltage-gated channels open. So this triggers our channels. Sodium rushes in, which depolarizes the membrane. And then the sodium channels close, the potassium channels open. Potassium rushes out, repolarizing the membrane and slightly hyperpolarizing it beyond resting membrane potential. And then our pumps reset us back to resting. Okay, basic action potential steps. So just to make sure we're all really clear on the vocabulary, although hopefully we covered some of this before. So the threshold is the voltage that activates the voltage-gated ion channels. And it actually activates both the sodium and the potassium channels at the same time. But the sodium channels are fast to open in response to the stimulus, and the potassium channels are slow. And that's why they open later, which works out really well. So the fast sodium channels are going to get us that depolarization, the super fast positive charge switch inside the membrane as the sodium floods in. And then because they're basically on timers, then the sodium channels close. Once the potassium channels open, the potassium floods out, and that gives us our repolarization. So now we're reversing our charge back to negative inside of the membrane. So when we say that action potentials are all or none, what we mean is that action potentials occur in the exact same way every single time. If we hit threshold, we get the exact same action potential every single time. The only way to not get that exact same one is if it doesn't happen at all. So if we don't hit threshold, we don't get an action potential. Okay, so that's what all or none means. A couple other things. So hyperpolarization is that overshoot when we become more negative than resting membrane potential as a consequence of the potassium flooding out. And then the refractory period, remember, is the time when the cell cannot be stimulated to have another action potential. What we didn't talk about before is that there's actually two parts to the refractory period. There's an absolute refractory period where no stimulus, no matter how strong, can stimulate an action potential. And then there's the relative refractory period where a higher than normal stimulus can actually trigger another action potential, but a normal one can't. So here's our more detailed action potential chart, right, where we actually have the little steps thrown in. So here we've got resting membrane potential, depolarizing stimulus to threshold, and then we trigger the voltage gated channels here. So then we have sodium flooding in, depolarization, potassium flooding out for repolarization and hyperpolarization, and then we're resetting here. And when we look at where the refractory periods are for that, we see that the absolute refractory period starts as soon as we trigger an action potential. It sounds kind of silly, but it means that we cannot trigger an action potential while another action potential is occurring. So unlike all of that muscle contraction that we looked at, where we could have an additive effect and get more and more force, we cannot add action potentials together. They either happen or they don't. And one of the reasons is that once we trigger those sodium channels, we can't re-trigger them until they finish resetting. And then you can see that once we drop back down to resting, we enter the relative refractory period. So during that reset, the sodium channels will be responsive. So it is possible to trigger another action potential here if we have a strong enough stimulus. Otherwise, we have to wait until the relative refractory period is over before a normal stimulus is able to trigger another action potential. So now we're going to contrast that with graded potentials. As soon as I tell the dog to stop barking again, because this is my life now. Okay, so graded potentials, very, very different from action potentials. So whereas action potentials are all or none, and they're the exact same way every single time, graded potentials are local, they don't go very far, they don't last very long, and graded just means that they can vary in strength. So remember that we had graded contractions when we were looking at motor units, where the stronger the strength of the stimulus, the stronger the response of the muscle, right? Up until a maximum. So graded potentials work very similarly. And the other thing that they can do is that they can be depolarizing, so we can have the membrane become less negative, but we can also hyperpolarize and actually make the membrane more negative, which is also not something that happens in an action potential, right? With an action potential, it always goes depolarize, repolarize, hyperpolarize, period. But graded can go either way. And part of that whole short distance thing is that the depolarization or hyperpolarization is going to be the strongest at the center where the actual stimulus hits, and then it weakens and disappears. Okay, so decay of signal strength. So here's a chart showing you stronger stimuli will cause stronger graded potentials. And that they can be depolarizing or they can be hyperpolarizing. So we can actually get the membrane more negative than resting with graded potentials. They're always going to be triggered by a stimulus. And remember, a stimulus is just defined as any change in the neuron's environment. But it's going to result in the opening of a gated ion channel. And so if the ion channel, lets like positive ions into the cell, then we're going to get depolarization. But sometimes it can let positive ions out, or it can let negative ions in, and that's how we get the hyperpolarization. Either way, we're going to call them a postsynaptic potential, because this is typically when we see this stuff. We're going to have a neurotransmitter crossing a synapse, binding to its receptor, and that's going to cause a graded potential. So this is typically what occurs between two neurons. Now, of course, there's more to it than that, but we'll get to that later. Okay? So here's another one showing you basically the same thing, because I found it and then I realized we had the chart in the book. But this one's nice because you can see the strength of the stimulus causing a variable strength in the response. So a little bit on how this spreads and why it decays. So if your stimulus occurs here, either because you're in a lab and you threw some electricity at it, or because there's a synapse here and we have neurotransmitter opening some channels, if you depolarize just this little bit of membrane, here's what happens. So you're going to have the positive charge spreading as it's attracted to the neighboring negative charge, and you're going to have the positive charge up here spreading or moving inwards as it's attracted to that subsequent negative charge. So we have this little swirling, they call it local currents of charge movement, which is basically movement of ions. Okay, so we're having diffusion of ions, which is going to spread the change in the charge. But that's only going to last so long, right? So because we're going to run out of ion movement, because we're not opening additional channels, we're going to have a slow decay of either depolarization or hyperpolarization, depending on what's actually happening. Okay, so we lose signal strength because, we only had so many ions, moving through the membrane here. All right, so that's why they're very, very local, because unlike the stuff we've learned about before, we're not triggering additional channels to open. We're just having localized changes in ion composition, which changes the localized charge. So the reason we have both is because graded potentials are what happen to receiving neurons as a response to stimulation. Now, if enough graded potentials depolarize the cell, enough of the cell to threshold, we can actually get an action potential. So action potentials are always going to be the output of a neuron when stimulated all the way to threshold because of the graded potentials. And if that doesn't make sense, I've got a couple more slides that hopefully will explain it. The video link is just a super cute graded potentials versus action potentials if you want to watch it later on. I also have this comparative chart for you so that you can better understand the difference between the two of them, because they are both important and necessary for the function of neurons. So I just showed you how graded potentials can only go so far and they lose signal strength. But we know that one of the important features of an action potential is that it's able to maintain the exact same signal strength over long distances. So the ability of action potentials to do that is called propagation. And the reason why it works is because of those same voltage-gated sodium and potassium channels that we learned about when we looked at muscle impulses. So basically, once we start an action potential, it's going to depolarize the membrane enough to trigger adjacent voltage-gated sodium and potassium channels, which causes another action potential. This causes the signal to move down the axon towards the axon terminal. We cannot have movement of action potentials back the other direction. And this is because of the, oh gosh, hold on, give me one, sorry. Jesus. This is because of the refractory period. So the refractory period prevents backwards movement of the signal. This is literally why we have a refractory period. So we never have a neuron sending out an action potential and then getting anything back up its axon. It does not happen that way. Not in nature, at least. We could artificially trigger one, but we don't want to because it wouldn't know what to do with it. All right. So it only receives stuff on the dendrite side from other synapses, and it only sends out information down the axon. So here's a little GIF kind of showing you that. So you can see it's receiving stimuli here. If the whole membrane up to the axon hillock depolarizes to threshold, we're going to trigger an action potential. And then we're going to get depolarization because sodiums come in, and then we get repolarization because potassiums go out, and then we reset and go again. Okay? I know there's a little chaotic, but it was kind of cute and it was the best I figured I could find. So the key here is the axon hillock. The axon hillock is the only part of the neuron that can initiate an action potential. And that's because it has a really high density of those voltage-gated channels. So once it depolarizes to threshold, we activate the voltage-gated channels. We have an action potential at the axon hillock. So here we have the beginning of it. The beginning of it triggers the next section. And so as this guy is finishing its action potential here, this guy is starting it. And now this guy's in refractory period, the initial segment. This one's finishing its action potential, but it triggered the next section, so this one's starting. So even though we talk about action potentials as if it's a single action potential moving down the axon, what it actually is a series of action potentials moving down. You can think about it very much like dominoes. One knocks down the next, right? It's not one domino that makes it all the way to the end, but it starts with one domino and everybody else goes because of it. So this movement down the entire axon with one segment triggering the next, it's called continuous conduction, okay? So propagation, continuously down the axon, continuous conduction. So the reason the signal doesn't die is because we're restarting it every single time, and because action potentials are all or none, we always maintain the signal strength. Now, this process of continuous conduction is fairly slow because it takes a couple milliseconds for every single action potential to happen, right? It's all or none. It always happens the exact same way. And, you know, we got to trigger the next one to get the next segment. So that's where myelinated fibers come in. When we want a faster signal, we myelinate the fiber. So when the axon is myelinated, what happens is there are no voltage-gated channels in the segment that's myelinated. Now, luckily, it's not luck, it's entirely science, but the nice thing is that the segments are set up so that a single action potential will depolarize the membrane the distance of a myelin segment. So we have voltage-gated channels here that are going to get triggered to open because we have an action potential here. So action potential here activates this. We get an action potential here, which activates this, and so on and so forth. So instead of continuous conduction where every single segment depolarizes and has an action potential, we're actually jumping down the membrane one myelin section at a time, and only the gaps are having action potentials. So this saves us a ton of action potentials and actually speeds things up a lot. Because it's jumping, we use the term saltatory, because apparently that means jumping or skipping. And that's why there's a rabbit right there too, is actually a jackrabbit. So this is much, much faster because we save a ton of time because we've decreased the number of action potentials that are being used. And that leads me to my next point, which is conduction velocity of action potentials down axons. So because we have this variability in the speed of nerve impulses, not all axons actually conduct nerve impulses at the same speed. And that's okay because not all of them have to be fast, okay? Some of them can be slow. Sometimes there's no rush. Oops. So there's actually two factors that determine the speed of an action potential down an axon. So impulse propagation, rate of impulse propagation is the same thing as conduction velocity. It's how fast we're getting it down the axon. One of them is degree of myelination. So not all axons are myelinated the same. So sometimes the entire length of the axon is myelinated, but sometimes only parts of it are. Obviously, more myelination is going to get us faster conduction velocity, right? Because we're skipping more segments of membrane. The other thing that determines rate of propagation is the diameter of the axon. So a larger diameter fiber has faster nerve impulses, and this is because of a surface area to volume ratio. Surface area volume. So when you have a small diameter fiber, more of the cell is membrane that's depolarizing versus when you have a large diameter fiber. And so it's able to go faster on the large diameter fiber. So that means that we have a whole range of conduction velocities in the nervous system. Large diameter, heavily myelinated axons are the fastest. And so signals that have to be fast for our survival are going to be carried on large diameter, highly myelinated axons. When it doesn't matter how fast something goes, then we might see it being carried on a small diameter, unmyelinated axon, because that's as slow as it goes, right? That's all continuous conduction, and it's the slowest type because of the diameter, okay? But we also have everything in between, because we can have smaller diameter but well myelinated ones. We can have less myelinated fibers where the whole length isn't necessarily myelinated. And all of those things will give us an entire range of conduction velocity from, you know, whatever, however slow it, I don't know it off the top of my head, it doesn't really matter, however slow our small diameter naked ones go versus how fast our large diameter heavily myelinated ones go, okay? Any questions about any of that stuff? No? Okay. So I'm gonna leave us here for now because I think that's as much as I can say without starting to make more pronunciation errors and forgetting more words. So we're gonna pick this up on Monday. We'll talk about synapses and neurotransmitters, and then we'll start talking about the central nervous system. Okay. So let me stop the recording and then I'll restart it and we'll do a little bit of, just to make sure everybody's on the same page about the, you know what I mean, lab.